Okay. Thank you for the introduction, Nico, and thank you all for organizing the seminar. My name is Qiuyue Chen, and I'm a postdoc from John Dobley Lab. Today, I'll share some work or recent work on quantitative genetics of maize domestication, where we found that a conserved genetic architecture among populations of the maize progenitor Teosinte was radically altered by domestication. So first, let's take a brief look at how Teosinte and maize look like. From the picture, you can see there are striking differences in plant architecture, where Teosinte has many long branches, and each branch has bending small ears and terminated by a tassel, while maize plant has a single stalk with a large or with one or two large ears. So this striking difference is mainly controlled by a gene called TB1. And also there are profound and complex differences in ear architecture. So for Teosinte, the ear has only 2 rows and including 5 to 12 kernels, and each kernel is enclosed by hard fruit case, while maize ear has at least 4 ranks and has a few hundred to hundreds of kernels, hundreds of naked kernels, and the kernels are attached onto the cob. So this dramatic difference is mainly controlled by a gene called TGA1. The evidence is from phylogenetic analysis and archaeological record both supported that maize arose by a single domestication from Teosinte in southwestern Mexico about 9,000 years ago. And the wild ancestor of maize is from a specific type of Teosinte called Pavignomis. So how did the genetic architecture of maize domestication look like? In 1939, George Bieder, the Nobel Prize winner of one gene, one enzyme, nailed out the Teosinte hypothesis, where he suggested that the origin of maize is from Teosinte. So later in the 1970s, after his retirement, Dr. Bieder designed a mass genetic experiment where he grew 50,000 maize Teosinte F2 plants, and he observed that 1 in 500 plants was either like the maize or the Teosinte parent. By doing the math, he concluded that 4 or 5 genes could account for the transition from the original Teosinte to maize. And in 1990s, John and his colleague mapped the QTLs for plant and inflorescence architecture trees in 2 independent maize Teosinte F2 populations. And by summarizing the QTL effects over 9 traits, they confirmed Bieder's hypothesis that a few large effect loci in about 5 or 6 regions could control the key morphological changes during maize domestication. So several of these large effect genes has been cloned subsequently. I also want to mention that during maize domestication, there is about 18% nucleotide diversity reduced during maize domestication. And one important research area for scientists is to mine and utilize the not so favorable layers from Teosinte, from the wild Teosinte, and apply it to the modern maize breeding. Beyond the genomic changes, studies also show that gene expression and there is strong divergence for gene expression and the metabolites during domestication. So our research questions are to use the classical quantitative genetics and trying to answer 3 main questions. The first one is, how did the genetic architecture among populations of Teosinte influence domestication? The second one is, how did the genetic architecture among populations of Teosinte was altered by domestication? And the third one is, how did the genetic architecture change within populations of Teosinte and maize? To answer the first two questions, our lab published a paper 2 years ago where they used a large sympatric population pair of maize and teosinte to study the genetic architecture of maize domestication. In this study, they used 50 teosinte parents to create about 4,500 progeny, and also used 50 maize parents to create the same number of progeny for comparison. So as shown in this map, the orange color shows where the previous study sampled the population, which is in the state of Mexico, right at the maize domestication center. To broaden the research questions, we further sampled 4 additional sympatric population pairs of maize and teosinte in 4 states from Mexico. And in each population, it includes 10 parents for both maize and teosinte. The principal component analysis shows that there is strong divergence between maize and teosinte, and also among populations within teosinte and maize. So next, we self and outcross the 10 teosinte parents to create about 1,250 progeny for each population, giving a total of 5,000 progeny for the 4 teosinte populations together. We took the same strategy for maize landrace and produced about 5,000 progeny for the 4 maize populations. And the progeny of maize and teosinte was then grown in Florida in 2 neighboring fields, and the phenotypes were then scored over 2 field seasons. To summarize our genotypes and phenotypes, so we have 4 populations for teosinte and maize respectively, and both of them have around 50,000 GBS SNPs. And we scored a total of 16 traits. And these traits have been classified into 3 trait groups by their genetic relationships. So the first trait group is vegetative or flowering time traits, which include such as days to anthisis, days to silking, and plant height. And the second trait group is environmental response traits, for example, the tenor number. And the third group is reproductive traits, such as earnings and green weight. With the genotypes and the phenotypes, we first performed a univariate analysis in the population. We fit a univariate mixed linear model in SRAMO and use the restricted maximum likelihood to estimate the variance. So in this model, the fixed effects include such as the year effect and the field coordinates. And the random effects include the additive effects, dominance effects, and additive by environmental interaction effects. So by solving this model, we can obtain the additive genetic variance, dominance genetic variance, G by E interaction variance, and plus the environmental variance, we can get the phenotypic variance. So first, how was the additive genetic variance affected by domestication? By plotting the proportion of phenotypic variance for both teosinte and maize, we can find that there is a higher level of heritable additive variance in teosinte than maize. especially for the reproductive traits. This suggests that the additive genetic variance is depleted during domestication. And then by plotting the proportion of genetic variance, which is the sum of additive and dominance variance, we can find the genetic variance is shifted to dominance. Again, especially for the reproductive traits. So the above results are from the combined 4 populations. We also estimate the variances among the four small teosinte populations and the 4 small landrace populations, and we got the similar results. So next question is, how much have traits diverged? So we plotted the log2 of the trait value in maize divided by Teosinte, and we can find that there is strong divergence for reproductive traits and environmental response traits, but not too much for vegetative or flowering time traits, especially the days to anthysis and days to seokin. While among the environmental response trait group, the Tiner number shows the most divergence. Among the reproductive traits, the grains per year shows the most divergence. Given the phenotypic divergence, we want to know, are the traits differences due to selection or drift? So we performed the QST-FST test to compare the neutral marker divergence to the trait divergence, where the FST measures the portion of genetic variation at a neutral marker that is between populations, while QST measures the portion of quantitative phenotypic variation that is between populations. So if the QST is significantly greater than FST, then we infer the selection drove divergence for the traits. So our results show that selection drove divergence strongly for the reproductive traits and also environmental response traits, but weaker for the vegetative or flowering time traits. Again, we also performed the QST-FST test in the 4 individual pair of Teosinte and maize population, and we got a similar result. So beyond the QST-FST between Teosinte and maize, we also performed the QST-FST test within Teosinte population or within maize population. And here, each dot stands for QST value in a comparison, and the red color indicates significant QST from the FST. So from this plot, we can see that selection drove changes in basically all traits between Teosinte and maize, but selection is far less important for explaining domestication trait differences among populations within Teosinte or within maize. Next question is, how strong was selection during domestication? We applied the univariate breeder's equation to calculate the selection intensity. So in this equation, the R stands for the response to selection, and I is the selection intensity. And H square is the natural sense heritability, and sigma P is the phenotypic standard deviation. By using this equation, we calculate in our population that the selection intensity ranges from 0.03% to 2% per generation. In this plot, the purple distribution shows the from the published studies of the natural selection, and the results, the selecting intensity from our traits are shown here. And we found that artificial selection is generally weaker than the natural selection. And among the 3 trait groups, the reproductive traits show stronger selection than the other 2 groups. Again, we also calculate selection intensity in each individual pair of Teosinte and maize population, and basically get a similar result. But there do have some variation. For example, in population 1 and 2, some of the reproductive traits has very large selection intensity. So next, we perform the multivariate analysis, which is multiple trait level. So we fit the bivariate mixed linear model in SRAMO, and also use the REML to estimate the genetic correlations between traits. In this model, we simplified compared to the univariate analysis due to computational speed. So we only include the additive effects as the random effect. So by solving this model, we can obtain the additive genetic correlation between traits, and we can also obtain the genetic variance covariance matrix, which is short for G matrix. These 2 diagrams show the examples of positive and negative genetic correlation between the 2 traits. So the causes of genetic correlations are mainly linkage or pleiotropy, and the consequences of genetic correlation can cause genetic constraint or indirect selection. With the genetic correlation matrix and also the variance for each trait, we can easily obtain the G matrix. So first, let's look at the additive genetic correlations among traits in our population. So the lower triangle represents the genetic correlation matrix in Teosinte, and the upper triangle represents the genetic correlation in maize landrace. So we used a test called Mantel test to test the correlation between the maize genetic correlation matrix between Teosinte and maize. And so for the full matrix, we can see the correlation is about 0.5, which is significant. So we conclude that the genetic correlation between Teosinte and maize are conserved. And we also performed a test for each submatrix for each trait group, and we found they are even better preserved, especially for vegetative or flowering time traits, followed by reproductive traits. Again, we also performed we also calculated the genetic correlation matrix in each individual population and did the Mantel test. And so in each of the single population, we also observed that the genetic correlations are conserved overall, and also better conserved in some submatrices. So in addition to compare the genetic correlations between Teosinte and maize, we also compared the genetic correlations among the Teosinte populations. And we can see the correlation is even larger than between Teosinte and maize, especially for the reproductive traits. The correlation is nearly 1 for the comparison. So this means that the genetic correlations are very conserved among Teosinte populations. And we also did the same thing among maize populations. overall they are conserved, although not as high as among Teosinte populations. So the genetic correlation structure or gene matrix can affect the rate and direction of evolution. So the evolutionary goal, the eventual evolutionary goal is to reach an optimum between traits. So if the two traits has negative correlation, then the path to reach the optimum will be slow and twisted. And if the two traits has positive genetic correlation, then it will be faster to reach the optimum. So now we ask, did the gene matrix pose constraints to domestication? Presuming maize is the optimum, and here the black arrow indicates the vector of evolution or domestication, and the theta t is the genetic is the angle between the genetic correlation structure and the vector of domestication. So if the theta t equals to 90 degrees, then we see the genetic correlation structure has full constraint to domestication. If the theta t equals to 0 degrees, then there is no constraint. And by calculating the theta t in our population, we found that the theta t is equal to 66 degrees at the initial domestication, and after domestication, the angle becomes 80 degrees. So we concluded that the gene matrix poses considerable constraints to domestication, and it increases over time. So how would the gene matrix affect domestication? Here shows again the univariate breeder's equation, where the R is response to selection, and S is selection differential, and H square is the narrow sense heritability, and the H square is equal to the additive variance divided by phenotypic variance. So then we can easily transfer this equation to the multivariate breeder's equation, where R, so here R is a vector of selection responses, and G stands for the G matrix, and beta is a vector of selection gradients. By using the multivariate breeder's equation, we ask, has the genetic correlation structure changed between maize, Teosinte, and maize? So we used a test called random squares test to test the response for Teosinte and maize, to test the similarity between the two gene matrices between Teosinte and maize. And so this test, basically simulated, we simulated 10,000 randomly selecting vectors, and then compare the response from two random gene matrices and with those response between Teosinte and maize landrace. So under null hypothesis, the correlation between Teosinte and maize response is no different from the correlation between response calculated from two random gene matrices. So the test results show that the gene matrix of Teosinte and the gene matrix of maize have been altered overall. And among the three treated groups, the vegetative flowering time correlations were conserved, and environment response correlations were also conserved, but the reproductive correlations were strongly altered. Again, we performed the test in the 4 individual pair of populations, and we got a similar result. So, in addition to the randomized score test for the geometries between Teosinte and the maize population, we also compare the geometries among Teosinte populations. And you can see they are, like for Teosinte, they are all significant. That means the geometrics or genetic correlation structures are very conserved among Teosinte populations, especially for reproductive traits. And among maize populations, with a few exceptions, the geometries look conserved. So next, we ask what traits are causing constraints. Basically, we performed a drop-one analysis. So in this figure, the Z is a vector of evolution, and Gmax is a vector of genetic correlations. So we already calculated the angle between the Z and the Gmax. So now we drop one trait at a time and calculate the angle again. And if the theta drop one is smaller than the theta Z, then it says that this trait increases constraint. And if theta drop one is larger than theta Z, then we say this trait decreases constraint. So by calculation, and we found that Florentine trait correlations constrain the evolution, and the reproductive correlations assisted evolution. Again, we also analyzed for individual population pair with only one exception in POP1. This natural branch node number trait. The other, generally, they are consistent with the combined 4 populations. That Florentine constrained evolution while reproductive traits assisted evolution. Another question we asked is like, what is the quickest path from Teosinte to maize? We already mentioned that multivariate selection with genetic correlations will have consequences of constraint and indirect selection. So again, this is a multivariate breeder's equation, and delta Z stands for the response vector, and here is the geometric, and beta is a vector of selecting gradient. So what we did is to simulate a hypothetical selection differential actor, which is a vector like the vector has an element 1 for a single for each trait, and 0 for all the other traits. So in this case, we can evaluate. So the trait with the element 1 is direct selection, and all the other traits will be indirect selection. So the question is like, if you apply selection to just one trait, like which trait provides the most gain along the domestication trajectory, given the genetic correlation structure? In this plot, again, the Z is the vector of evolution or domestication, and the Z is the so the R is so the R is a response for each trait by using the multivariate breeder's equation. And we calculate the angle between these two vectors, and we also project this vector onto the Z, onto the vector of domestication to project it onto the vector of domestication. And by doing this, we found that the grains per year, cupules per rank, or year length provided the most gain along the vector of domestication, while the flowering time provided the least evolution gain along the domestication trajectory. Again, we performed the same analysis in the 4 individual populations, and basically they are consistent with what we observed for the 4 combined populations. So now let's take a brief summary of what we observed and get from all the analysis. The genetic architecture of maize domestication can reflect the following results. First, there is depleted additive genetic variance during domestication, especially for reproductive traits. And the second is genetic correlations or G matrix offer strong constraint for domestication, with flowering time contributing most to the constraint. And genetic correlation structure for reproductive traits have changed strongly. Also, the artificial selection was weaker than natural selection, and reproductive traits were the primary target of selection. Last but not least is that selection drove divergence for most traits between Teosinte and maize, but is far less important for explaining trait difference among population within Teosinte or maize. Finally, I'd like to thank all our co-authors for this project, especially my advisor, John, for all the supervising and insight for science questions, and also Jim and Peter for excellent statistical guides, and also CJ and Fernando for setting the basis of this project. So my work is next using more populations on their workspaces. So thank them all. Okay, that's all. With that, thank you